you guys had a little bit of fun getting dressed up and doing some stuff up on stage last Friday, and so I thought that I would actually have a little fun myself. And I was unable, as you know, to decide which section had actually done the best job. So I decided that you would all get a reward. And in order to understand the reward, I want to give you the background, which actually comes from a very, of course, deep theoretical investigation of the kind that I always want you to know about. Right? So let me get this up there. Here we go. I want you to understand the Swiss theorem, okay? And the Swiss theorem is really a very essential part of population biology. Basically, it tells you that the partial derivative of happiness with respect to an increase in chocolate intake is positive across the entire reaction norm of the human condition. Okay, so chocolate makes you happier, right? And the Swiss, as is usual, claim priority for something which is in fact a contribution of another major civilization, which is the Mexicans. Now, you will notice that I have had the teaching fellows divvy up the chocolate onto paper plates and distribute it in small quantities around the room. This is because the homogenization of fitness within groups promotes group benefit and causes a general increase in group success, because the only way then that the group can improve is that if the performance of the individuals increases the performance of the whole group. Now, that's about all that I really want to say, except that I want to thank you very much for participating in that exercise last Friday. I thought it was interesting. I enjoyed it. I hope you've had a chance to look at it. And the only issue that now faces me is whether I should continue to give the lecture in this costume. I have been told by my teaching fellows that I absolutely have to do it, right? But I have difficulty believing that you'll believe me if I look like this. Will you believe me? You want me to leave it on? All right, let's go. Okay, so we're actually now going to go back to, oh, by the way, there is a pedagogical message in this costume, and that is, if you can't play like a child, you can't be creative. Okay? It's about playfulness. Okay. Have you all got your chocolate? Are you happy? Or let me put it this way, are you happier? It looks like we have some leftovers. And so you're all welcome to grab some on the way out. The basket, by the way, is a working basket from a market in the Ivory Coast, and when people make baskets to work with, they make them really well. So I don't want the basket to disappear, okay? Now for today's lecture. What we're looking at now is the impact of space on communities and on the distribution of plants and animals around the planet. So I'm going to talk today about island biogeography and metapopulations, and I want you to start thinking of the world as fragmented, okay? So it's spatially heterogeneous. There are islands, there are mountains, there are lakes and oases, and a lot of the world is becoming fragmented by humans. So one of the big ways that humans are having impact on the planet and changing life on the planet is by fragmenting the landscape. So the issue today is, what is determining biodiversity in a fragment? What happens when we break the world up? Here are some sketches of what happened in Cadiz Township, Wisconsin, between 1831 and 1950. So before the settlers go out, we had an eastern hardwood forest, and as the area got settled, this is 1882, 1902, 1950, you can see the forest disappears and there are little blocks of trees that are left as forest fragments scattered across the landscape. And this is where the birds and the rodents and the coyotes and everything else now, and the deer, have to try to make their living. And if we now look at Wisconsin from space, so this is in the early part of this century, you can see a picture which actually has cities in it in red and a landscape which is largely agricultural with some little dark blocks of forest scattered through it. So this is mostly agriculture here. This would be forest. Let's take another look. Another kind of natural fragmentation, of course, is archipelagos, islands. And now we're getting towards island biogeography. This is an observation made by Robert MacArthur and Ed Wilson that plots on the y-axis, on a log scale, the number of species on an island, and on the x-axis, the area of the island in square miles. And this is for the Sunda Islands, so this is in Indonesia. And up here, you've got the Philippines and New Guinea added to the Sunda Islands. And what you see is that on a log-log plot, the bigger the island, the more the species. And this is the part of the world we're talking about. Komodo dragons come from Komodo here. The little dwarf Homo erectus was found on Flores. This is Timor. New Guinea is going to be off here somewhere. Java is over there. And Krakatoa is down here. So these are the Sunda Islands. And they have lots of different surface areas. I mean, if you just look at this little one and compare it to this big one, you're going to have a lot fewer birds on there than you are on there. So in trying to come up with a general theory of biogeography, MacArthur and Wilson did the standard Cartesian analytical reduction of saying, what are the essential features of that system? What are the fewest things that we have to pay attention to that will tell us something, a take-home message, some key message that we can pull out of the system? And they thought, well, let's suppose that there isn't any evolution going on, and that all of the species we're considering already exist on some big continental landmass, and that they're getting onto islands in a process of immigration, and they're dying out on islands in a process of extinction. And this is the number of species that are present on the island, okay? So they argued that in such a situation, the number of species on the island will come to an equilibrium. There'll be species that are coming off of the continents, they're flying, drifting, getting blown, whatever, out onto the islands. And the immigration rate will start high. When the island is empty, everything that shows up on it is a new species. But the immigration rate must inevitably fall down to zero when the number of species on the island equals the number in the source pool on the mainland. Okay? Because we're just counting species. We're not counting number of individuals arriving. I mean, if 100 birds fly in and their species already exists on the island, that doesn't count for immigration because that species is already there. So that's the way this axis is constructed. So this goes down and this goes up. The number of species on the island is going to be affecting the extinction rate probably in two ways. The simplest is, the more species there are, the greater the chance that one of them will go extinct, just at random. So that curve is going to go up just because there are more species on the island. However, if there are interactions among the species on the island, such that predation, disease, whatever is going to drive one to extinction, you can see that it might bend upwards. So it's not just going to be linear, it's going to go up. So that's how they argued the curves. And they said where the curves intersect, the number coming in will equal the number going out, and that's the number we should expect to find on the island. So far, so good. This is all just a priori. Well, what's going to affect the rates of immigration and extinction? Well, first, they argued that immigration will decrease from islands that are near the mainland out to islands that are far from the mainland. So you could construct a series of curves. So this would be for the curve for an island close to the mainland, and this would be the curve for an island far from the mainland. And that's just because it's harder to get out there. On the other hand, they argued that extinction rates will increase as you go from large islands down to small islands, basically because there's more space on the big island, more different niches, more places, habitats where organisms can live, more different kinds of things could survive there. But also, as you get onto a small island, the intensity of the biotic interactions are going to get bigger, and it's going to be harder to get away from a predator or harder to get away from a parasite. And so you could imagine that as you shrank an island, extinction rates would go up. So they predicted, hey, we could have an equilibrium over here on an island which is small and far from the coast, or that we could have it here if it was a small island close to the continent. And similarly, if we have a large island which is far away from the continent, we might be down here, and a large island close to the continent would be here. A big one close to a continent, by the way, would be Trinidad. Trinidad has almost the biodiversity of neighboring Venezuela. A small one, far away from a continent, so with a very low biodiversity, might be something like, you would think, an isolated oceanic island like Easter Island or Hawaii. So, why is this important? Well, for a long time, from the mid-1960s up to about 1985, 1990, this was the only game in town. And there weren't alternative ways of thinking about these processes, and it played a big role in the design of natural parks and nature reserves. Essentially, it said, because of the effective area on biodiversity, it's better to have a big park than a small one. And because of the effect of immigration rate on biodiversity, it said it's better to provide corridors to connect landscape fragments if you possibly can, so that things can immigrate and move back and forth. So it was used a lot. However, when we summarize it, you'll see it's an equilibrium between colonization and extinction. It assumes there's a source population, so evolution isn't going on. There's a source population out there. That's where all the species are. There's no speciation occurring on islands. There are just two effects that you're worried about: how big the island is and how far away it is from the mainland. Extinction is driven by area, and colonization is driven by distance. Okay? Now, all of these things that I've just written down here will be important for you to remember if you are asked how to reconstruct the equilibrium number of species on an island in a certain circumstance. Okay? I want to emphasize that. It's the kind of thing that might turn up on a midterm. If you're on a small island a long way from the mainland, you'll have low species diversity, and a big island close to the mainland will have high species diversity. And that seems to be an intuitive point, but I've given you an analytical framework from which to derive it. Now, as you'll see in a minute, I am now going to blast this theory out of the water. I'm going to take it apart and show that it makes a whole series of assumptions and claims that are demonstrably not true. And before I do that, I want to signal to you that I'm going to come back after I do that and say, hey, it was still a good thing. Okay? So, the theory is dealing only with the number of species, not with the number of individuals. There's no population dynamics. I mean, if you've got 10 of them on the island or 1,000 of them on the island, you count them the same way. That seems to be a little silly, because the probability of extinction should be related to the number that are there. All of the species are considered together. It's just kind of one general immigration rate and one general extinction rate. You know, it's kind of fun to wave my arms when I've got this gown on. But the probabilities of immigration and extinction are different. I mean, it's going to be different for birds and ants and mosses and paramecia and elephants and stuff like that, okay? So they must some way differ systematically. The island biogeographic theory, kind of like the Hardy-Weinberg theory, it's an equilibrium theory, okay? It doesn't allow for history. But if we just look at what's happened in the Western Pacific in the last 10,000 years as the Polynesians came out and colonized those islands, we know that 25% of the birds that were on them have already gone extinct. So you send your budding biogeographer out there to get a sample from the Sunda Islands or from Guam or from the Philippines or Micronesia, and they come back with a count of the number of bird species on the island. It's a very misleading count, because 25% extinction has already happened on those islands. And that's where those data came from that I showed you. Okay, this effect has not been compensated for in that data set. Theory doesn't allow for speciation and adaptive radiation, and you take one pair of cardueline finches from Central America and fly them out to Hawaii 15 or 20 million years ago, and voila, this is what they produce, okay? Bunches of species. So that's going on, and that's not in the theory. It assumes that the probability of being able to immigrate doesn't depend on how many species are already there, okay? But the presence of some species is probably a prerequisite for that of others, and the presence of some may keep others from coming in. So those are probably effects to worry about. It's actually empirically difficult to decide when immigration has occurred. So you're sitting here out in the Thimble Islands, okay, off Brantford, Connecticut, and it's spring, and a bay-breasted warbler comes flying through. Do you count it? Well, it's going to just stop off, eat a few insects, and fly on to Canada. It's just passing through. Is that immigration? Probably not. So you have to actually find the species that breed on the island. That's not so easy. We assume the system's at equilibrium, but how can we recognize an equilibrium when we've got one? There's not a clear prediction on how fast this turnover would occur. Or do we have to wait 10 generations, 100 generations, 1,000 generations? And hey, what about the problem that generation time is fast for little things and slow for big things? That makes that issue pretty complicated. Then if it is at equilibrium, the assumption is that every time a new species comes in, one that was already there goes extinct. Well, that seems to be a little unrealistic. That's a very tight coupling of immigration and extinction, and the real relationship's weaker. So, the major assumption of the theory, which is that there's a turnover of species that produces an equilibrium between immigration and extinction, is correct. That's been tested experimentally on small islands. But the observed turnover is often of casual species, not the ones that breed with established populations, and there's nothing in the theory to tell us what proportion should be in each category. So, the theory is a failure. The theory is a failure if the goal is to be right. Okay? But hey, the goal is not to be right. The goal is to try to explore nature in such a way that we discover the truth. And the theory is a great success if that's what we want to know. We can only operate with a working hypothesis. For a long time, this was the only game in town. And the criterion of success is how much work gets stimulated by the idea. Okay, you'll see shortly that a lot was stimulated by it. And how rapidly can it be constructively falsified and replaced with a better theory? The pages of science are littered with the corpses of dead theories. And sometimes a theory has the delightful adaptation that it contains within itself the seeds of its own destruction. And the seeds of destruction of a theory are often how fascinated people get by it and how hard they're willing to work to try to test it. And that's what happened to island biogeography. It was a good one. So we can look at MacArthur as a Dionysiac, a creative enthusiast. We can look at Mark Williamson as an Apollonian objective critic. And if you like that dichotomy, then you ought to go back and read Friedrich Nietzsche's The Birth of Tragedy, which he wrote when he was still a PhD student and only 23 years old. That was long before, by the way, he went crazy. Okay, so that's one view of the universe. That's the island biogeography view of the universe. Now I want to do metapopulations because this is the other major alternative way of looking at spatial dynamics of species and populations. So, a metapopulation is a set of local populations that are linked by movement. And as with island biogeography, the dynamics of metapopulations are driven by extinction and recolonization, or immigration. So I'm going to be talking a bit about some of these species. Okay, frogs, butterflies, thyme plants, pathogen populations living in us, things like that. So here's the basic conceptual framework. Here we have a local population, and it's got reproduction going on in it. It's producing an excess of organisms, and they're moving out into the environment because it's getting crowded locally and they want to find a place to live. They go out and they can find an empty patch to colonize, and sometimes, for one reason or another, their population will go extinct in a local patch. So if you just take a big sample of patches across the landscape, each of which is a population, you will find some of them with thriving biology going on, and some of them are empty, and they can be empty because they went extinct, or they can be empty because they were never colonized. and all of them have the conditions that the organisms need to survive in. So if you build a simple metapopulation model, you can pull out some pretty important, straightforward messages. One of them is this: even if every single local population is likely to go extinct, the metapopulation can survive in a balance between extinction and colonization. So basically, you should think of it as, oh my heavens, we're about to go extinct locally, I'd better get up and fly out and go find a new place and just keep hopping. And if a population manages to do that, it can keep itself going even though it leaves behind a long trail of consistent extinction. The landscape is important in this, and of course that's very attractive because it gets people into landscape ecology, it gets them into photographs taken from space, it gets them into geographic information systems, and it's actually created, that whole area is now a new field of analysis. So the landscape features that are going to affect extinction and colonization then are going to be things that are very important for regional persistence. So, you know, if we're dealing with, say, Daphnia living in ponds in Connecticut, and in any particular pond in Connecticut, Daphnia is likely to go extinct, but we look across the whole state, we see that there are 100,000 ponds. Daphnia actually ends up probably doing just fine in Connecticut. And if you doubt that, I suggest you just take a beaker of pond water, not city water. City water's got a lot of chlorine in it. Take a beaker of good natural pond water and put it on top of your residential college in downtown New Haven and go back 6 months later. You will find in it rotifers, algae, and copepods, and they will have fallen into it out of the air because these guys fly around in the air. You might not think so, but they manage to get up there. Another message is that there is a ratio between colonization and extinction above which a metapopulation can exist. So if you're concerned with the question, is there an analytically determinable threshold for our Daphnia population in the state of Connecticut that will tell us how much they have to move around in order to stay here in the long run? The model will give us that, okay? That's the threshold ratio between colonization and extinction, and it's kind of a simple number. It tells us those are the rates we have to worry about. And you can interpret that in terms of the proportion of patches that are occupied and average patch size. So there's actually something you can go out and measure that will give you an estimate of this ratio, which will tell you, will the thing persist or not? So if you're concerned with population viability analysis, if you're concerned with conservation and with the threats to biodiversity, this is something that you can actually go out to measure and then construct an argument with. And you can back your argument up with a literature that has now some rather impressive logic in it. So here are a few insights. It's perfectly normal to have some local empty patches. You know, in my village in Switzerland, they were very worried about their carabid beetles because that's all they had left. And they had some carabid beetles, they had a few toads, and they had some salamanders. And there wasn't very much left in the forest. And locally, people would get very desperate about their local pond not having any salamanders in it anymore or something like that. Whereas if they would back up and they would look at, say, a chunk of landscape that was 100 kilometers on a side, they could relax because local things are often going extinct but then being recolonized. And you actually need to be able to back up and look at it at a fairly large spatial scale and a pretty long time scale before you can discern the overall trend. This places a big demand on data collection, but it leads to a lot more realism in making forecasts. So you need to look at the region of the landscape rather than the local population, but there is something that's hard to measure, and that's the migration rate. Okay? It's just hard to see. After all, if I were a salamander, I would do it on a rainy night, right? At about 2 o'clock in the morning, and who's going to be out there tracking me around? So this is hard to measure. Is there evidence that, in fact, nature is organized this way? Well, we know that population size is significantly affected by migration, and we can see that in both the source effect and the sink effect. So you put a fence around a population, and if it's a source, it will increase, and if it's a sink, it will disappear. And that can only happen if The source would normally be exporting migrants, immigrants that are going out, and the sink would only happen, it would only disappear when the fence is put around it if it had been previously maintained by stuff coming in from sources. So that's experimentally demonstrable. We know that population density is affected by the area and the isolation of the patch. Big patches tend to have slightly higher densities, and distant patches tend to have lower densities. If it's really a metapopulation, then the population density should be going up and down out of synchrony. Okay, if it's really tightly linked and there's a tremendous amount of immigration, then you can just treat the thing as just one big population. But if it's a metapopulation, and some things are doing okay and some things are going extinct, then doing okay means going up, and going extinct means going down at the same time. So they would be asynchronous, and that is often observed. Is there population turnover? Do local populations go extinct and then get recolonized from a source? And that's been observed, at least in one case, for snails living in ponds in the UK. And that's done by taking samples of the mud in the bottom of the ponds and going back over many years and looking for the presence of snails. And they disappear and they come back, and they disappear and they come back. If you're a good naturalist and you know where your beast likes to live, then one often sees that suitable habitat is present, but it's empty. And both in a plant population and in a butterfly metapopulation, both of these metapopulations persist despite lots of local extinction. So the butterfly population is in Finland, and the plant population is in Provence. It's time in Provence. And both of these, by the way, if you like metapopulation biology, you get to do lots of neat field biology and do it in wonderful circumstances. And since the French use the thyme in their cooking and the butterflies are beautiful, this is a nice sensory experience as well. The risk of extinction in a metapopulation does depend on patch size. If you're in a small one, it's much more likely that you'll go extinct than if you're in a large patch. So the evidence for that's pretty strong. And the colonization rate depends on patch isolation, and that's true for most species. I would like to note here that if you take the Max Planck Gesellschaft A330 up for a little tour around the globe at 35,000 feet, and you put out a plank, you slow the plane down enough so you can put out a plankton net, and you trawl for aerial plankton up in the stratosphere, you will find baby spiders and fern spores covering the planet up in the stratosphere. You can do that over the South Pole at 35,000 feet, and you will find, in a state, believe me, of deep hibernation, little tiny baby spiders rafting along at minus 70 degrees Celsius, and they're still alive. Fern spores will do the same thing. So in fact, these are exceptions. The colonization rate, depending on patch isolation, however, would be very important for elephants, rhinoceroses, bears, stuff like that, okay? So you can see that there's a gradation based on dispersal ability, body size, lots of biology. Small isolated patches are likely to be empty. Big connected patches are likely to be full. Lots of evidence for that. That's certainly a straightforward prediction of the model. And a fugitive competitor can exist in a metapopulation. A fugitive competitor is one that if you just put them into a local equilibrium population, they will get beaten out by the other species that's there. But if they are better at dispersing while the other one is better at competing, they can keep jumping out and getting ahead of that. And that's been well investigated with Daphnia in the Finnish archipelago. Furthermore, I'm going to show you a shot of a prey species that would go extinct in a local population, but it can coexist with its predator in a metapopulation. And I'll show that for two mites living in greenhouses. Now, the point that is made here is that when you shift from the equilibrium local population perspective up to the metapopulation perspective, the complexities of spatial distribution will allow a lot more things to coexist with each other. And that's true both for competition theory and it's true for predation theory. So, here's the Finnish archipelago. It is, by the way, continuing to emerge from the water. because there's glacial rebound. After the Pleistocene glaciers melted, they had depressed the underlying continental crust, and it is now rebounding and rising up. So these islands continue to come out of the water. And they are a lovely kind of fairy tale kind of landscape, filled with all sorts of interesting biology. They have on them, for example, 6-foot-long water snakes that are about that thick. And they have lots of birds and other things. And on them, they have little pools of fresh water, surrounded by this part of the Baltic, which is not really seawater. It's kind of brackish. But the Daphnia can't really survive in the sea. The salinity is still too high for them. So they move around among pools on these islands in places like this. This little island here might have 10 or 20 pools on it, and they are moving around among these islands, probably having their ephippia, which are their resting stages, born on the feet of shorebirds that are flying. And there are at least 2 species of Daphnia that live in the Finnish archipelago. One of them is a better competitor, one of them is a better disperser, and they coexist because what one lacks in competition ability, it makes up in dispersal ability. So if you have that kind of a trade-off, you can generate a persistent metapopulation in a system like that. I strongly recommend, by the way, if you're ever in Stockholm or in Helsinki, that you go out into the Baltic archipelagos. They are just beautiful landscapes. The other is mites in a greenhouse. And this is Carl Huffaker's experiment. And he had a brilliant idea for a model system in which you could investigate the impact of spatial structure on predator-prey interactions. He had a herbivorous mite that likes to eat oranges, and these are oranges, okay? And he had a predatory mite that eats the herbivorous mite. And what he did was he constructed a model ecosystem that consisted of a whole bunch of oranges interspersed with billiard balls. Well, obviously, the herbivorous mite can't eat a billiard ball, but it can eat an orange. And then he altered the migration rates of the species by putting grease down in between the two. So he had a system that you could actually put in your kitchen cabinet that had a complete spatial ecosystem in it, and he could play with the parameters. And what Huffaker discovered is that he could have persistence of a predator and prey with spatial structure where if they were confined to a single orange, they would both go extinct. First the prey would go extinct and then the predator would starve. Okay, so back to a comparison between the 2 ways of looking at the world. If you look at the number of publications per year using, say, Web of Science, you can see that interest in island biogeography peaked in the mid-'80s and then has declined. It's not gone to 0, but it's going down. But since 1985, there's been an explosion of interest in metapopulations. And there are really 2 reasons for that. One of them is that it is obvious that the landscape really is fragmented. And so metapopulation theory has become an organizing concept in conservation biology, where people try to maintain biodiversity at a landscape scale. We can see it all around us. It's much easier to study and manipulate a metapopulation than it is to manipulate an archipelago. So there's been a lot more progress doing experiments like the one that I showed you that Carl Huffaker did. That was actually before the theory came up. He was sort of a prophet way ahead of his time. And there's an analogy to epidemiology, and it's quite compelling. And we know that epidemiology works. So I now want to show you the analogy with epidemiology. And this is a connection now between ecology and infectious disease. So the host is a local patch. Here's a local patch. That looks like a good one to infect. Here's another one. Boy, am I going to get her in the dorm, you know. Pathogens have local populations within hosts. Now, what would constitute extinction? Well, extinction would be either you kill your host, so you die with it, or the host develops an immune response. Okay, so you can go extinct for either reason. And by the way, if you're a pathogen, either is equally bad. The disease transmission rate is equivalent to the migration rate. And if we look at that, I'm now going to go back and I'm going to re-rehearse this issue of measles on islands and in big cities. It was mentioned last Friday, briefly. But it's an important example, and I feel entirely unashamed about mentioning an important example twice. There might even be a better chance of it being remembered a week or 20 years later. So measles in big cities are a huge metapopulation with a continual input of young hosts that don't have any immune defense. Babies, okay? Luscious, little, susceptible babies, ripe for infection. On islands, an island is a tiny little metapopulation. You know, look at the island, look at the Falkland Islands or the Orkneys or something like that, Pitcairn Island, Easter Island. Very few hosts, and if measles could get onto an island like that, or any other infectious disease that causes a sterilizing immune response, as measles does, then it'll sweep in a wave through that island, and everybody will become immune before enough babies can be born to maintain the disease. So repeated extinction occurs. Here is the incidence of measles in big cities and on islands between 1921 and 1940. So this is before measles vaccine, when you could study this as a natural process. And 0 years with a month of no cases in the big cities. And as we go from fairly large islands down to smaller islands, we have more and more months with no cases until you get to the Falkland Islands. And over that 19-year period, there wasn't a single case of measles in the Falkland Islands. They must have been pretty worried about a ship coming in that had somebody with measles on it, but there was no case during that 19 years. So here's a guy with measles. Here's the pathogen. This is the situation in a big city, and here is the Falkland Islands. As you can see, the density that you have in a big city just makes for wonderful transmission possibilities. Fantastic. So diseases will tend to go extinct on little islands, and host populations will then lose both their acquired and their inherited resistance. And if then after many years the disease is reintroduced, the epidemic can really be catastrophic. So I think you already know that on Hispaniola, that's the Dominican Republic and Haiti, between 1492 and the late 1500s, a population of about 500,000 indigenous Americans was reduced to 300 by measles and by other diseases. Also, when the conquistadores landed at Veracruz and started marching on Mexico City, the epidemic preceded them, so the Aztec army was being decimated by disease when they got there. But you know, the Aztecs really weren't, it's not the only explanation. The Aztecs were not very well liked. This habit of ripping people's hearts out and eating them on altars hadn't endeared them to the captives that they got and the subject peoples. And so actually it only took 900 conquistadores to defeat the Aztec army because they had 200,000 local allies that said, yeah, we want to beat them up too. So it was both that effect and the disease effect that allowed the conquest of Mexico. In a city, what's going on is that the pathogen population is being rescued by the colonization of empty habitat. So this is the rescue effect in a metapopulation, and it's the rescue effect in epidemiology. And that basically, the pathogens are being rescued by babies. And the babies are born susceptible. They do not yet have an acquired immune reaction. They haven't built up the population of cells that will target that particular pathogen. And so that rescues the disease before it goes extinct. And there are enough of these babies coming in a city so that the colonization rate, the transmission rate, and the number of occupable sites is high enough to keep that population going. So the take-home point on this lecture, besides the fact that the professor is crazy and wearing a mask, is that geography is very important in ecology, and there have been a number of pretty big attempts to create analytical systems to deal with it. A lot of the world is fragmented. I think even the abyssal plain is getting fragmented as trawlers start going deeper and deeper, and if we undertake mining operations in the North Pacific to pick up little modules of molybdenum and things like that, we're going to just continue to disrupt the entire planet. Movement by organisms among fragments creates a dynamic across the whole landscape, and local extinctions and recolonizations may be an entirely normal thing, and you can't really see that until you look at a big enough chunk of space in a long enough period of time to establish the metapopulation dynamic. And then finally, I'd like to emphasize that the spread of disease, epidemiology, can be viewed as a metapopulation dynamic and can be viewed as a model system within which to test metapopulation assumptions, and when you do that, it seems to work pretty well. Okay, so next time I'm going to talk about the flow of energy and matter through ecosystems, and if I can get my costume off, I am available to go to lunch today.